Hi everyone, I'm sorry I couldn't be there today. My name is Yiraldi Loera and I'm going to be presenting the historical and ecological histories of Hirunda rustica. So I wanted to quickly outline the presentation so you could get a sense of the flow of ideas from the identification of Hirunda rustica to their life histories and then their behavior, which is dictated by their phylogenetic relationships, and their overall species biogeography, which is also affected by population genetics and responses to environmental change, which is going into my research. So Hirunda rustica, as you may know, are also known as the barn swallow, and these are very charismatic birds with a bright cobalt blue dorsal side and a beige to rust-colored ventral side. They have long tapered wings for agile flight and a very distinctive tail. This tail is so special because it has characteristic tail streamers at the ends to help the swallow stop very quickly or turn rapidly, and they're also very good indicators for health. So these are model species for natural selection studies as well as sexual selection studies, as they're really good ornamentation features preferred by females. So this is a migratory species with breeding in the north as seen in the light beige, and they migrate towards the equator or southern hemisphere during the winters as seen in the dark brown. They're found globally except most of Australia and Antarctica, and avoid the Saharan desert because of its temperatures and also because there are less human settlements there. They live in grassland habitats, primarily feeding on insects as an aerial forager. And they build their nests on anthropogenic buildings, which I'll further discuss later, but they are of low concern on the IUCN list. And quickly, this is a clip of the high-pitched chirps and calls. So briefing you very quickly on the taxonomy, Hirunda rustica are in the kingdom Animalia, phylum Chordata, class Aves, as you guys may all know. Order is Passeriformes, and family is Hirundinidae. So Passeriformes are the passerine birds, or the perching birds, which are identified by their 3 unwebbed toes in the front and a very strong and flexible hallux in the back that lets them perch easily on tree branches. And so when not perching or flying, they are in or creating their distinctive mud nests. So what they do is they scoop up mud or manure mixtures with their mouths and mix it with saliva to form these nests, mainly stationed on anthropogenic buildings or barns, giving them the name barn swallows. And they nest in pairs and avoid large colonies due to high instances of parasitism and also competition. So going into one of the studies, they presented evidence on the relationship between bar swallow phylogeny and their behavior of mud nest building. So they superimposed nest data on a DNA hybridization phylogeny of 17 swallow species, and they showed that nest construction is tightly linked and is tightly linked to the inferred evolutionary history, with burrowing as a primitive mode of nesting, giving rise to cavity adopting and mud nesting clades. And this mud nesting clade has evolved once in history and has been increasing in complexity through time. So within the species of Hirunda rustica, there's also been a recent switch from natural nest sites to human structures, as proven by a different study. And they conducted phylogenetic analysis using mitochondrial DNA and nuclear DNA to explore the recent evolutionary history of the species, and they found that mitochondrial DNA does show a recent split. However, the nuclear DNA did not show any phylogenetic splits, meaning that the main clades, being Africa, Europe, Asia, and the Baikal region and the Arctic or North America, have evolved recently. And so a little bit of history on this. After the separation from an African sister taxon, the barn swallow achieved more of this fully Arctic distribution, and then a vicariance event happened where there's a separation of the Western Palearctic, the European clade, and then another vicariant divergence of the Asian and North American clades. And interestingly, there was a secondary invasion from North America to the Baikal region, And even though dispersal is not ongoing, they still have this charismatic red coloring on their ventral side, either indicating stabilizing selection or insufficient time for divergence. So a more recent study just confirmed this phylogenetic relationships of these subspecies and their origins, which corresponds to different vicariance events shown V1 through V3, and a dispersal event from North America to the Baikal region. And so this phylogeny corresponds to 6 closely related subspecies with a nearly worldwide distribution due to their close proximity to humans. And this graph is actually created or modified by Dr. Scordato, the population genetic professor here on campus. And it shows the subspecies distributions and the recolonization from North America to the Asian Tylary. So with Dr. Scordato's help in population genetics, I was actually able to study the subspecies Hirunda rustica from populations in Africa and the Middle East. So I wanted to determine if there was population differentiation within the subspecies in these areas and why. So it can be due to the fact that phenotypic traits under selection are associated with genome-wide differentiation, meaning there's reduced hybridization and symmetry when they're together. It may be due to sexual signals leading to differences in sexual signals leading to reproductive isolation and reduced gene flow, or overall morphological adaptations that lead to population differentiation. So Dr. Scordato gave me a data set with 227 individuals, 5,000 SNP loci, and 10,000 possible alleles that are codominant from 8 sampling sites across Africa and the Middle East. And we used R and a couple population genetics software programs to determine basic diversity statistics on the population, including FST values, which are values of differentiation. And we analyzed this differentiation by clustering analyses and outlier detections of variable SNP loci. Very quickly, some of the results showed that there was subdivision within Hirunda rustica populations across Africa and the Middle East, and this coincided with geographic location and the subspecies, as shown on the colored graph, and on the x-axis of the principal component analysis. And despite it being only a shallow differentiation with only 6 outlier loci variable between them, it was further subdivided by sex as seen on the y-axis of the principal component analysis. So future tests would further rule out hybridization within these populations as they have yet to diverge into distinct species, and we still don't know exactly what led to this subtle differentiation, but Dr. Scordato suggests that it may have to do with reproductive isolation due to differences in sexual selection, since it is further subdivided by sex. And so sexual selection plays a strong role in these species, in the biogeography of these species, especially with their tail streamers and plumage coloration. So plumage coloration actually plays a role in survival, and pheomelanin plays a role in this colorization as it causes the yellow or red pigmentation of the hair and feathers. And studies have shown that this is also advantageous in many environments, as the more pheomelanin a bird has, the greater the residual annual survival rates. And this is interesting to ecotoxicology, which is my favorite field to study, as pheomelanin is directly affected by oxidative stress. So oxidative stress is when there's an excess of free radicals that overcome the power of the body's antioxidant system, and it causes a stress situation. And the stress can lead to a slew of maladies from the brain to the skin and its pheomelanin. So my lab researches the effects of oxidative damage caused by ionizing radiation. And one instance of exposure to ionizing radiation are nuclear disasters. So, for instance, studies in Chernobyl have shown that barn swallows are one of the 57 breeding bird species in the area that are affected by ionizing radiation. And the study also showed that they're pretty good bioindicators of other species' health, as they have a nearly cosmopolitan range and are ideal migratory species, but it was also shown that their abundance also decreased through time and increased in effect as radiation levels increased. So upon inspection, especially in the high radiation levels, they did display albinistic feathers, and this is a clear sign of oxidative stress due to the lack of phenylalanine. So this case of albinism is also seen in Fukushima, with the frequency of partial albinism being around 2 times the control, just by simple surveys by my professor and his colleagues. And these abnormalities have been shown to be heritable but not yet studied, since Fukushima is a more recent disaster. So I've been working on a meta-analysis of studies including natural disasters, nuclear plants, and other types of exposures to ionizing radiation to determine the best detection method of mutations for this phenomenon to be tested of being by germline origin. So from 221 studies on the effects of environmental radiation, ionizing radiation on mutation rates, we found that chromosomal aberrations are best at determining a high effect at nuclear accidents and nuclear plants, followed by specific gene mutations using mini-satellites, microsatellites, or more importantly, or more specifically, SNPs. SNPs are much more sensitive to detecting mutations, so they are ideal in detecting specific gene mutations. And so we hope to apply these techniques on parent and offspring pairs of pairs collected using DNA samples from Fukushima, and we hope to determine if this abnormality is of germline origin. And this effect may be altering their species biogeography and their population genetics overall, so it's a pretty interesting field that covers this pretty multidisciplinary field. So this is the end of my slideshow, and thank you.